So in the last part of chapter 10, we're going to examine what happens when the cell cycle breaks down. So we're going to talk about cancer and its relationship with the cell cycle. We need to talk about how cancer is caused, and it's really uncontrolled cell growth, but we'll talk a bit about the reasons behind that, or some of them at least. We need to talk about these things called proto-oncogenes. These are normal genes, and once there are mutations in them, we will call them oncogenes. So we'll talk about those. And then we need to talk about tumor suppressors again. And we'll talk about their role in preventing cancer, and when they break down, that can lead to cancer. All right, so cancer cells are different from usually the cells around them. They often have mutations that lead to breaking the cell cycle regulation. This often has side effects. They grow bigger, there's more of them, they look weird, they often have all kinds of issues with their chromosome number. So we'll talk a little bit about this, but this is very generic. There are many, many different types of cancers. There is not one disease that is cancer. There are many causes. There are many ways that it can come about. But there are some common mechanisms that we tend to find in most cancers. Cancer, in and of itself, is uncontrolled cell growth. That's the definition of cancer right there. Even with all the checks, mutations are going to happen. So one of the unfortunate things about being a eukaryotic animal is that if you live long enough and your cells divide enough, mutations will occur. Okay? And that means that if you continue to live longer and longer, your chance of getting cancer goes up. That's even if you don't have any factors contributing to it. So you don't encounter any mutagenic chemicals or things like this. Mutations just happen because when the DNA copies itself, it makes mistakes. It is not 100% accurate. So even with all these checks and all these things, if you live long enough, you will get cancer. And that's why we see cancer rates go up as you live longer. The probability gets higher and higher. The accumulation of mutations in cells, particularly in cell cycle and reproduction genes, increases that chance of cancer. So there is a genetic component to this. If you already have a few mutations, that means it takes less to end up with a potential cancer there. So cancer risk goes up with age because of those more mutations. Now, there are, of course, things that can increase your chance of cancer. But when somebody says, oh, that's going to give you cancer, that's not really quite accurate. It might increase the chance of getting cancer. That's kind of the way it works. So we need to define a mutation here. Mutation is not growing a third arm or glowing or being anything like that. Mutation is simply a change in the DNA sequence that may or may not affect a gene. And in many cases, when genes get mutations in them, changes in their DNA sequence, they tend to not work anymore. So with that knowledge there, let's talk about how cancer can arise. So we have a group of genes that generally are positive regulators. These are positive regulators of the cell cycle, and their job is to push the cell through the cell cycle. We would call these proto-oncogenes. Most mutations that happen in these positive regulators mean that they can't do their job. The cell can't divide. No damage is passed on. But I said in the last section that sometimes there are mutations that might cause these positive regulators to become stuck in an on position. We would now call these things oncogenes. CDK is an example of this. Remember, we said CDK levels are constant, but they don't function without that cyclin binding to it, unless there are mutations that lead to CDK thinking that it has cyclin bound to it, and that would keep the CDK on constantly. So there are actually several cancer treatments that target CDKs. So here are our cyclin-dependent kinases. You can see these drugs. I'm not going to attempt them. I'm too tired to try to read drug names right now. You can see these all work on cyclin-dependent kinase 4 here. So there are numerous drugs that work on CDKs. Now, this is just one example of a problem with a positive regulator. There are many, many more that are there as well. We also have breaking the tumor suppressor genes. So remember, the tumor suppressor genes were negative regulators. When they break, that means that there's nothing to stop the cell cycle from progressing. The genes were called tumor suppressors because that's their normal function. When they're broken, tumors might result from this. We talked about a few of those in the last section, but P53 is one of those key ones. P53 blocks the G1 checkpoint. But we have sequenced the genomes of many, many cancers, and we find that in about 50% of all human tumor cells, there is a mutation in P53. Okay? That means that in the other 50%, other stuff is broken, but P53 is so commonly associated with cancer that it is one of the first things that we would look at to see if that has happened. Normally, P53 is checking for any sort of DNA damage and will lead to fixing that, but if it can't, it sends the cell into apoptosis. If that breaks, right, the cell cycle is continuously on. So here's P53 in its normal state. It's leading to cell cycle arrest, DNA repair, restarting the cell cycle if it can fix it. Or if it's like there's too much damage, that cell is destroyed. But when P53 has mutations in it, there's nothing to stop the cell from moving on to the next state. Oftentimes, this is going to lead to uncontrolled growth and division, cancer. So why is the coordination of the cell cycle so important? Well, let's look at some karyotypes here. Here's a normal human cell karyotype. You can see that there are 2 of each chromosome, one from the maternal and one from the paternal lineage. Here are some cancer cell karyotypes. You can see there are 1, 2, 3, 4, 5, 6 copies of each of these chromosomes here, and little bits of the red chromosome have been stuck onto the yellow one here. This one has 3 of every chromosome. This one's had a big translocation there. This one has had little bits that have broken off and moved around. These are all highly abnormal, right? That's because the proper cell cycle was not maintained. So let's think about this. Let's do a little review question. The cancer cell karyotype pictured had some sort of issue with the cell cycle checkpoint. What checkpoint do you think it had an issue with? Go ahead and pause the video. Think about this. There is not necessarily one correct answer here, but the most likely answer, in my opinion, would possibly be G2. My argument for that would be G2 is making sure that the DNA was replicated correctly. If we don't stop there, and we have improper replication of the DNA, then we might get duplicates of the chromosomes here. There is another possibility, though. Could also be the M phase. So remember, in M phase, we're checking that the spindles are attached properly. Oftentimes, when they're not attached properly, both of the chromosomes get pulled into one cell and the other one gets none. And that can lead over time to these duplications of chromosomes. So I gave you a little bit of curveball there. This is just more of a thought question here, but you could even maybe argue that G1 has failed, right? Because that checks that there's no damage in the DNA before going into the cell cycle. So there's kind of multiple possible rationales here, but I would bet G2 in this case. Okay, so that's our crash course into cancer. Cancer is not caused by one thing. It is uncontrolled cell growth, and there are many ways that that can happen. It's usually caused by mutations which break cell cycle regulators. So there are lots of different regulators, both positive and negative, and mutations in those can lead to cancer. Now, luckily, there's a lot of redundancy in this system. I will show you later how much DNA repair actually occurs, but over time, mutations will accumulate. I want to reiterate, a mutation is just a change in the DNA sequence. That is the definition we will use in this course. I expect you to know that definition. You need many mutations for this uncontrolled cell growth to occur. So that's usually why we see cancer becoming more common the later you are in life, because you've accumulated more mutations as you've lived. In positive cell cycle regulators, we call them proto-oncogenes. When they're doing their job, they're just sending the cell through the cell cycle when it needed to. But if they get broken and stuck on, we call those oncogenes, things that constantly send the cell into division. Negative regulators, on the other hand, we call them tumor suppressors normally because they stop the cell from dividing. But when they are broken, there's no checks on that cell cycle, so the cell can keep going through the cell cycle there.